Hi class, this is your instructor, Scholar Huff. We're now in chapter 19. This is evolutionary change in populations. Each population possesses a gene pool. So keep in mind that again, evolution occurs in populations, not individuals. You do not evolve. I do not evolve. Individuals are not where evolution occurs. It is only at the population levels. So again, as this happens, each population possesses a gene pool, which includes all the alleles for all the loci present in the population, and it's because diploid organisms have a maximum number of two different alleles at each genetic locus. And if you've forgotten the term locus, which I mentioned a moment ago, the term loci, it is the location of a gene on a chromosome. So as I was saying, at each genetic locus, a single individual typically only has a small fraction of alleles present in a population's gene pool. So the genetic variation that is evident among individuals in a given population indicates that each individual has a different subset of alleles in the gene pool. The evolution of populations is best understood in terms of genotype, phenotype, and allele frequencies. So what we're doing here is getting to the term called population genetics. So as we get here, it will help you develop an understanding of the important genetic variations as the raw material for evolution and the basic concepts of population genetics, which is the study of the genetic variability within a population and the evolutionary forces that act on it. So population genetics represents an extension of Mendel's principles of inheritance. So let's get down into it. I've already got to the term gene pool, so let's further ourselves into terms. Genotype frequency. So when I mention this, it is the proportion of a particular genotype in a population. And I don't really want to use these huge numbers here, but let's look at, if you can, in your brain, four different individuals. One individual is homozygous dominant. One individual is homozygous recessive. Two individuals are heterozygous. So it would be 25% homozygous dominant, 25% homozygous recessive. And then 50% of those individuals are heterozygous. So there you have that genotype frequency. And I can use what's being presented here, but that's just a lot of genotypes there. So I gave you percentages, so I guess I should say it will be 0.25 would be the homozygous dominant and homozygous recessive genotype frequencies, and then 0.5 for the heterozygous. Next up is phenotype frequency. So this is the proportion of a particular phenotype in the population, if each genotype corresponds to a particular phenotype. For instance, if I go to that very same example with the four individuals, 0.75 of those would exhibit that dominant phenotype, and 0.25 of those would exhibit that recessive phenotype, because there was one homozygous dominant and there were two heterozygotes, and then there was one homozygous recessive individual. Now to allele frequencies. So an allele frequency is a portion of a specific allele in a particular population. Now, counting all this up is going to be a little different. But back to the example I used moments ago, there would be 2, 4, 6, 8 alleles in total. So there is there are 3 recessive alleles. 3 recessive alleles. And since 3 of those alleles are recessive, 3 out of 8. That's some math to do in my brain, you all. Let me think here. That would be 0.4. So 0.4 for the homozygous recessive. And then, if that's 0.4, 0.6 for the dominant allele frequency. So the dominant allele frequency is 0.6, and the recessive allele frequency is 0.4. And again, there were 3 recessive alleles in my example, and there are 5 dominant alleles in my example, and the total number of alleles is 8. So I won't go through the examples given in the book at the moment, but you definitely can believe I have examples for you all. Hardy-Weinberg principle. So in what we did here, the book will take us back to booked examples. So what I'm getting at the moment is, by way of the Hardy-Weinberg principle, I'll say that the Hardy-Weinberg principle shows that if a population is infinitely large, the process of inheritance does not by itself change in allele frequencies. It also explains why dominant alleles are not necessarily more common than recessive alleles. So the Hardy-Weinberg principle represents an ideal situation that seldom occurs in the natural world. However, it is useful because it provides a model to help us understand the real world. So knowledge of the Hardy-Weinberg principle is essential to understanding the mechanisms of evolutionary change in sexually reproducing organisms. So if I go back just a moment here, and I'll pause right there. So in this example, we observe that 90 of the 1,000 individuals in the population exhibit the recessive phenotype characteristic of the genotype homozygous recessive. The remaining 910 individuals exhibit the dominant phenotype, and they are either homozygous dominant or heterozygous. So you might assume that after many generations, the genetic recombination during the sexual reproduction would cause the dominant allele to become more common in the population. And you might also assume that the recessive allele would eventually disappear altogether. These assumptions were common among many biologists in the early 20th century. However, they are not correct. They are incorrect. They are not correct, because the frequencies of alleles and genotypes do not change from generation to generation unless they are influenced by outside factors. So a population whose allele and genotype frequencies do not change from generation to generation is said to be at genetic equilibrium. So such a population with no net change in allele frequency or genotype frequencies over time, it is not undergoing any evolutionary change. So a population that is at genetic equilibrium is not evolving with respect to the locus being studied. However, if the allele frequencies change over successive generations, evolution is indeed occurring. So the explanation for this stability of successive generations in populations at genetic equilibrium was provided independently by Hardy, an English mathematician, and Weinberg, a German physician, and this was back in 1908. And they pointed out that the expected frequencies of various genotypes in a population can be described mathematically. And that's what we got here with this Hardy-Weinberg principle. So let's now get to a couple of equations. The top equation is for allele frequency. So the dominant allele is always going to be represented by P as in Paul, and the recessive allele will always be represented by Q as in Queen. So P plus Q equals 1. Period. End of story. And you saw this a moment ago with my example given to you, where I mentioned in my example that the recessive allele frequency was 0.4 and the dominant allele frequency was 0.6. Those do equal 1. So P was 0.6 and Q was 0.4. And given Q, you can always find P. For instance, at the bottom right, if you have Q, then you take if you have Q, 1 minus Q, there at the bottom left, equals P. I'll say it again, at the bottom left, given Q, and I hope you're taking notes, at the bottom left, given Q, meaning we have Q. And just keep that in your minds. We will always find Q first, as we work out a problem with this. So given Q, you are always able to find P. because 1 minus Q equals P. And the same goes if you're given a problem and you're given P. In that case, 1 minus P equals Q. So here we're at that other equation, and this is for genotype frequencies. So P squared equals the frequency of the homozygous dominant. 2 times P times Q equals the frequency of the heterozygous. And then Q squared equals the frequency of the homozygous recessive. And I say again, we will always find Q first, and the way to find Q first will be to first use what is Q squared. If we use what is Q squared and we take the square root of Q squared, we then are able to get Q. Given Q, we can then find P. 1 minus Q is P. And please rewind this if I'm going too fast. I apologize if I'm going a little fast. This can be found in your textbook on page 408. 408. So in this example, it says, Hardy-Weinberg calculations begin by determining the frequency of the homozygous recessive genotype. There again, it tells you to always find Q first. So we're going to begin with finding that Q squared. So it tells us here in this example that we have 90 homozygous recessive individuals in a population of 1,000. So the first thing that you should do is divide 90 by 1,000. Upon dividing 90 by 1,000, the answer is 0.09. That is Q squared. I hope you're writing this down. That is Q squared. So, as I mentioned a moment ago, and I hope I did not go too fast, but again, the first thing we do is, given the number of homozygous recessive individuals, we have our total number of individuals, divide part by whole. 90 divided by 1,000, which equals 0.09. That is Q squared. Note we're not quite done because we have to find Q first. That is Q squared. So given Q squared, we can find Q by taking the square root of Q squared. And that answer is 0.3. 0.3 is Q. So now that we have Q, there is nothing that the question can ask that you cannot answer. For instance, given Q, we can now find P. So 1 minus Q equals P. So 1 minus 0.3 equals 0.7. And there we have our answer. So now that we have both P and Q, we can now find everything else, meaning we can find the frequency of the homozygous dominant, we can find the frequency of the heterozygous there, and we were already given Q squared. That's where the problem began. So to find the frequency of the homozygous dominant, what we would do here is square P. So P squared is 0.49. To find the frequency of the heterozygous, we would now take 2 times P times Q. So 2 times P times Q would be 2 times 0.7 times 0.3, and that is 0.42. And we already have Q squared, and if you remember from back where we began, Q squared is 0.09. That is how you work the problem out. And I know I didn't work it out here. I don't have a touch screen computer, but I hope that you wrote this down with me because you will be doing this on your test. So back to Hardy-Weinberg, and this is the problem, but I never have worked it out this way. You will not work it out this way unless you, I don't know, but I never worked it out that way. So now, genetic equilibrium occurs if certain conditions are met. The 5 conditions are as follows. Mating has to be random. Mating has to be random. Number 2, there are no net mutations. There cannot be any mutations in the population. There is an infinitely large population. There is no migration. So there are no individuals coming into the population, and there are no individuals leaving the population. And then lastly, there is no natural selection. Those are those 5 conditions that must be met for a population to be, or at least meet that Hardy-Weinberg principle, to be in genetic equilibrium. So we're not going to do this with blood types. I left it in here and I considered it. I considered it. However, I would recommend you look through it on your very own time, because given what I did with that first example, you are now able to do this without my assistance. You have everything you need to do this. I'm getting now on to what is called microevolution and microevolutionary forces. So evolution presents a departure from the Hardy-Weinberg principle of genetic equilibrium. The degree of departure between observed allele and genotype frequencies and those expected by the Hardy-Weinberg principle indicates the amount of evolutionary change. This type of evolution, generation to generation changes in allele or genotype frequencies within a population, is sometimes referred to as microevolution. So it's all due to it evolving a relatively small or minor changes, usually over a few generations. So changes in allele frequencies of the population result from 5 microevolutionary processes. Know these for your test. It is non-random mating, mutation, genetic drift, gene flow, and natural selection. So I'll leave that there for you if you need those again. So these microevolutionary processes are the opposite of the conditions that must be met if a population is in genetic equilibrium. So when one or more of these processes act on a population, allele or genotype frequencies change from one generation to the next. Let's get down into it with non-random mating. When individuals select mates based on the basis of phenotype, they bring about evolutionary change in the population. So two examples are non-random mating, and two examples of non-random mating are inbreeding and assortative mating. In many populations, individuals mate more often with close neighbors than with more distant members of the population, and as a result, neighbors tend to be more closely related, that is, genetically similar, to one another. The mating of genetically similar individuals who are closely related or more closely related than if they had been chosen at random from the entire population is known as inbreeding. Although inbreeding does not change the overall allele frequency, the frequency of homozygous genotypes increases with each successive generation of inbreeding. The most extreme example of inbreeding is self-fertilization, which is particularly common in certain plants. Inbreeding does not appear to be detrimental in some populations, but in others it causes inbreeding depression. You should see your textbook, figure 19.2, on page 411. So in which inbreeding depression occurs, in which inbred individuals have a lower fitness of those not inbred, and fitness is usually measured as the average number of surviving offspring of one genotype compared to the average number of surviving offspring of competing genotypes. So inbreeding depression, as evidenced by fertility declines and high juvenile mortality, is thought to be caused by the expression of harmful recessive alleles because of homozygosity increases with inbreeding. This is one instance of such. This is figure 19.2. Please see this and hope that you understand this. You have in red non-inbred mice over time by way of weekly survival, and you have in blue those inbred mice over time by way of their weekly survival. So several studies have provided direct evidence of the deleterious consequences of inbreeding in nature, such as the white-footed mice, as you're seeing right here. So they were taken from a field and used to develop both inbred and non-inbred populations in laboratory. When these laboratory-bred populations were returned to nature, their survivorship was estimated from a release recapture data. This is ecology here. So the non-inbred mice had a statistically significant higher rate of survival than the inbred mice. It is not known why the inbred mice had a lower survival rate. Some possibilities include higher disease susceptibility, poor ability to evade predators, less ability to find food, and less ability to win fights with other white-footed mice. Assortative mating. So in which individuals select mates based by their phenotypes is another example of non-random mating. For example, biologists selected two phenotypes of high bristle and low bristle number in a fruit fly population. Although the researchers made no effort to control mating, they observed that the flies preferentially mated with those with similar phenotypes. Females with high bristle number tend to mate with males with high bristle number, and females with low bristle number tend to mate with males with low bristle number. Such selection of mates with the same phenotype is known as positive assortative mating, as opposed to the less common negative assortative mating, in which they mate with opposite phenotypes or select mutation. Variation is introduced into a population through mutation, which is the unpredictable or random change in deoxyribonucleic acid. So a mutation, which are the source of new alleles, result from 1. A change in nucleotide base pairs of a gene, or 2. The rearrangement of genes within chromosomes so that their interactions produce different effects, or 3. A change in chromosome structure. Mutations occur unpredictably and spontaneously. A particular locus may have a DNA sequence that causes certain types of mutations to occur more frequently than others. The rate of mutation appears relatively constant for a particular locus, but may vary by several orders of magnitude among genes within a single species and among different species. Not all mutations pass from one generation to the next. During those occurring in somatic cells or body cells, they are not inherited. When an individual with such mutation dies, that mutation is lost. Some mutations, however, occur in those germ cells or those reproductive cells. These mutations may or may not overtly affect the offspring because at least some of the DNA in the cell appears to be non-functional. So even if the DNA occurs in the even if the mutation, in the DNA that codes for a polypeptide, it may still have little effect on the structure or function of that protein. However, when the polypeptide is sufficiently altered to change its function, the mutation is usually harmful. So by acting against seriously abnormal phenotypes, natural selection eliminates or reduces to low frequency the most harmful mutations. Mutations with small phenotypic effects, even if slightly harmful, have a better chance of being incorporated into the population. Whereas sometime later, under different environmental conditions, they may produce phenotypes that are more useful or adaptive. So, a mutation by itself may cause small deviations in allele frequencies from those predicted by the Hardy-Weinberg principle. Although allele frequencies may have changed by mutation, these changes are typically several orders of magnitude smaller than changes caused by other evolutionary forces, such as genetic drift. Now on to genetic drift. So the size of the population is important in its effects on allele frequencies because random events or chance tend to cause changes of a relatively greater magnitude in a small population. So if a population consists only of a few individuals, an allele present at a low frequency in the population could be completely lost by chance. Such an event would be unlikely in a large population. For example, consider two populations, one with 10,000 individuals and one with 10 individuals. If an uncommon allele occurs at a frequency of 10% or in both populations, then 1,900 individuals in the large population have the allele. That same frequency in a smaller population means that only about 2 individuals have the allele. From this exercise, it is easy to see that there's a greater likelihood of losing the rare allele from the smaller population than from the larger one. Predators, for example, might kill the 1 or 2 individuals possessing the uncommon allele in the population purely by chance, so those individuals would leave no offspring. So let's get to the production of random evolutionary changes in small breeding populations is known as genetic drift. Genetic drift results in changes in allele frequencies in a population from one generation to another. One allele may be eliminated from the population purely by chance. Thus, genetic drift decreases genetic variation within a population, although it tends to increase genetic differences among populations. First up is the bottleneck effect. When bottlenecks occur, genetic drift becomes a major evolutionary force, and it's because of fluctuations in the environment, such as depletion of food supply and outbreak of disease. A population may rapidly and markedly decrease from time to time. The population is said to go through a bottleneck during the genetic drift can occur in the small population of survivors. As the population again increases in size, many allele frequencies may be quite different from those in the population preceding the decline. Scientists hypothesized that the genetic variation in the cheetah was considerably reduced by a bottleneck at the end of the last ice age, some 10,000 years ago. Cheetahs nearly became extinct, perhaps from overhunting by humans. The few surviving cheetahs had a greatly reduced genetic variability, and as a result, the cheetah population today is so genetically uniform that unrelated cheetahs can accept skin grafts from one another. Normally, only identical twins accept skin grafts so rapidly or readily. Next up, the founder effect. The founder effect occurs when a few founders establish a new colony. So when one or a few individuals from a large population establish or found a colony, they bring them only a small they bring with them, only a small fraction of the genetic variation present in the original population. And as a result, the only alleles among the descendants will be those of the colonizers. Typically, the allele frequencies in the newly found population are quite different from those in the parent population. The genetic drift that results when a small number of individuals from a large population found a colony is called the founder effect. The Finnish people may illustrate the founder effect. Geneticists who sampled the DNA from the Finns and from the European population at large found that the Finns exhibit considerably less genetic variation from other Europeans. This evidence supports the hypothesis that the Finns are descended from a small group of people who settled about 4,000 years ago in the area that is now Finland, and because of the geography, it remained separate from the other European societies for centuries. The subpopulation in eastern Finland is especially homozygous. It's especially homogeneous because it existed in a relative isolation after being established by only several hundred founders in the 1500s. There you have it. Gene flow. Individuals of a species tend to be distributed in local populations that are genetically isolated to some degree from other populations. For example, the bullfrogs of one pond form a population that is separated from those in an adjacent pond. Some exchanges occur by migration between ponds, but the frogs in one pond are more likely to mate with those in the same pond because each population is isolated from the other population. Each has distinct genetic traits and gene pools. The migration of breeding individuals between populations causes corresponding movement of alleles, or gene flow, that has significant evolutionary consequences. As alleles flow from one population to another, they usually increase the amount of genetic variability within the recipient population. If sufficient gene flow occurs between two populations, they become more similar genetically. Because gene flow reduces the amount of variation between the two populations, it tends to counteract the effects of natural selection and genetic drift, both of which often cause populations to become increasingly distinct. If migration by members of a population is considerable, and if populations differ in the allele frequencies, significant genetic changes can occur in local populations. For example, 10,000 years ago, modern humans occupied almost all Earth's major land areas except a few islands and Antarctica. Because the population density was so low in most small areas, the small isolated human populations underwent random genetic drift and natural selection. More recently, major migrations have increased gene flow significantly, altering the gene frequencies within previously isolated human populations. Now on to natural selection. So natural selection is the mechanism of evolution first proposed by Darwin, in which members of the population are more successfully adapted to the environment, have greater fitness. That is, they are more likely to survive and reproduce. Over successive generations, the proportion of favorable alleles increases in the population. So in contrast with other microevolutionary forces, natural selection leads to adaptive evolutionary change. Natural selection not only explains why organisms are well adapted to the environments in which they live, but it also helps account for the remarkable diversity of life. It is natural selection that enables population to change, thereby adapting to different environments and different ways of life. Natural selection results in the differential reproduction of individuals with different traits or phenotypes in response to the environment. Natural selection preserves individuals with favorable phenotypes and eliminates those with unfavorable phenotypes. Individuals that survive and produce fertile offspring have a selective advantage. The mechanism of natural selection does not develop a perfect organism. Rather, it weeds out those individuals whose phenotypes are less adapted to the environment, or at least environmental challenges, while allowing the better adapted individuals to survive and pass their alleles to their offspring. So by reducing the frequency of alleles that result in the expression of less favorable traits, the probability is increased that favorable alleles responsible for an adaptation will come together in the offspring. So now to adaptive evolutionary change. So to get here, we'll go through three kinds of selection that causes change in the normal distribution. So there'll be stabilizing selection, directional selection, and disruptive selection. So we'll begin with stabilizing selection. And when characters are under polygenic control, meaning a range of phenotypes occur, with most of the population located in the median range, and fewer at either extreme. So this arrangement is a normal distribution, which you see there on that left-hand side with no selection. That is a normal distribution. You may have heard this referred to as being a bell curve, but that is called a normal distribution. So three kinds of selection cause the changes in normal distribution, and although we consider each process separately, in nature their influences generally overlap. So with stabilizing selection, shown here at B is in boy, the process of natural selection associated with a population is well adapted to its environment. So most populations are probably influenced by stabilizing forces most of the time. Stabilizing selection selects against phenotypic extremes. It selects against phenotypic extremes. And in other words, individuals with the average or intermediate phenotypes, they are favored. So there's an example of this in your text. The blue curve shows the distribution of birth weights in a sample of 13,730 infant births. The red curve shows mortality or death at each birth weight. Infants with very low or very high birth weights have higher death rates than infants of average weight. The optimum birth weight, that is, one with the lowest mortality, is close to the average birth weight, which is about 7 pounds. So because stabilizing selection tends to decrease variation by favoring individuals near the mean of the normal distribution at the expense of those at either extreme. So the bell curve, in this case, it narrows. So although stabilizing selection decreases the amount of variation in the population, variation is rarely eliminated by this process because other microevolutionary forces act against a decrease in variation. For example, a mutation is slowly but continually adding to the genetic variation within the population. Directional selection. So if an environment changes over time, directional selection may favor phenotypes at one of the extremes of the normal distribution. Over successive generations, one phenotype gradually replaces another. For example, if greater size is advantageous in a new environment, large individuals will become increasingly common in the population. However, directional selection occurs only if alleles favored under the new circumstances are already present in the population. Darwin's finches provide an excellent example of directional selection. Since 1973, they've been studied, and the Galapagos finches, by their study by Peter Grant and Rosemary Grant from Princeton University, they did a meticulous analysis of finch eating habits and beak sizes during 3 extended droughts, one of which was followed by an extremely wet El Nino event. That was in 1983. During the droughts, the number of insects and small seeds declined, and large, heavy seeds became the finch's primary food source. Many finches died during this time, and most of the survivors were larger birds whose beaks were larger and deeper. In a few generations, these birds became more common in the population. After the El Nino event, however, smaller seeds became the primary food source, and smaller finches with average-sized beaks were then favored. So in this example, natural selection is directional. During the drought, natural selection operated in favor of the larger phenotype, whereas after the wet period, selection occurred in the opposite direction, favoring the smaller phenotype. I won't go to another example with bighorn sheep. I want to go now to disruptive selection. Sometimes extreme changes in the environment may favor two or more different phenotypes at the expense of the mean. That is, more than one phenotype may be favored in the new environment. Disruptive selection is a special type of directional selection in which there is a trend in several directions rather than just one. It results in a divergence or splitting apart of distinct groups of individuals within a population. Disruptive selection, which is relatively rare, selects against the average or intermediate phenotype. Limited food supply during a severe drought caused a population of finches on another island of the Galapagos to experience disruptive selection. The finch population initially exhibited a variety of beak sizes and shapes. Because only foods available on the island during the drought were wood-boring insects and seeds from cactus roots, natural selection favored birds with beaks suitable for obtaining these types of foods. Finches with longer beaks survived because they could open the cactus roots, and those with wider beaks survived because they could strip the tree bark to expose insects. However, finches with intermediate beaks could not use either food source efficiently and had a lower survival rate. And from here, I'll briefly go to genetic variation populations. Briefly. One thing I'll speak to is genetic polymorphism. So genetic variation among individuals in a population, it may or may not produce distinct phenotypes. So as I get to this, I'll go to a balanced polymorphism. And it's a special type of genetic polymorphism in which two or more alleles persist in a population over many generations as a result of natural selection. Heterozygote advantage and frequency-dependent selection are mechanisms that preserve balanced polymorphism. So, natural selection sometimes helps maintain genetic diversity, and in some cases, natural selection even may maintain the population's alleles that are unfavorable in the homozygous state. That happens when a heterozygote has a higher degree of fitness than the homozygote. This phenomenon is known as the heterozygote advantage and is demonstrated in humans by the selective advantage of heterozygous carriers of the sickle cell allele. The mutant allele for sickle cell produces an altered hemoglobin that deforms or sickles the red blood cells, making them more likely to form dangerous blockages in capillaries and to be destroyed in the liver, the spleen, or bone marrow. People who are homozygous for the sickle cell allele usually die at an early age if medical treatment is not available. Heterozygous individuals carry alleles for both normal and sickle cell hemoglobin. The heterozygous condition makes a person more resistant to a severe type of malaria than people who are homozygous with a normal hemoglobin allele. In a heterozygous individual, each allele produces its own specific kind of hemoglobin, and the red blood cells contain two kinds of roughly equivalent amounts. Such cells sickle much less readily than cells containing only the mutant allele. They are more resistant to infection to the malaria-causing parasite, which lives in red blood cells, than the red blood cells containing only the normal hemoglobin. Where malaria is a problem, each of the two types of homozygous individuals is at a disadvantage. Homozygous for sickle cell are likely to die of sickle cell anemia, whereas those individuals homozygous for the normal allele may die of malaria. The heterozygote is therefore more fit than either homozygote. In parts of Africa and the Middle East and Southern Asia where Falciparum malaria is present, Heterozygote individuals survive in greater numbers than the homozygote. So the mutant allele is maintained at a high frequency in the population, even though the homozygous recessive condition is almost always lethal. So this has been your instructor, Scholar Huff. Please study well, and thank you all for listening.